Alright guys, so welcome back. So the last section for talking about DNA is really kind of what we're culminating at. So now that we understand the basic structure of DNA, now we can start talking about how we replicate it. How do I make copies of DNA? And remember from last unit, the real reason we're making copies of DNA is because we're going through cell division. So if we remember our cell cycle here, right, G1 in interphase is our real purpose for growth, for development, for doing normal function. But once we pass that G1 checkpoint, everything from there on out is ready to prep yourself for mitosis. Or, if we're going through meiosis, that first round of meiosis 1. And DNA replication happens in that S phase, or that synthesis phase of your cell cycle, because we need to have enough DNA that when we get through a mitotic division and get through cytokinesis, those two new daughter cells have the same genetic information that the parent cell contained. So DNA replication, it's going to happen in the nucleus of your cell, and it's going to happen at these things called origins of replication. When we talk about replicating DNA in a prokaryote, so bacteria for example, those bacterial cells are only going to have one origin of replication. They're going to start at one spot and they're going to work through their entire single chromosome, duplicating their genetic information until they reach the other end. Eukaryotic organisms, however, something like us, where we've got 46 chromosomes that we need to duplicate before we can go through mitosis, if we started at one end of a chromosome and went to the other, and then moved on to chromosome 2 and started at one end and went to the other, started at chromosome 3, went to the other end, it would take forever. Okay? So as a way to speed up DNA replication, we're going to have these replication forks or replication bubbles or origins of replication happening in multiple places along a chromosome, and they're going to be happening in all 46 chromosomes at the same time. And that's going to help to speed up the replication process so that S phase doesn't take 4 or 5 years just to get us a new cell. All right, so we're going to zoom in to the single replication bubble and talk about what's actually happening there. So DNA replication, here is this replication fork, and this is one end of that replication bubble or origin of replication we were just taking a look at on the previous slide. And there's going to be a handful of enzymes you're going to have to be familiar with to explain how replication happens. And the first enzyme that's going to come in is something called DNA helicase. And DNA helicase's whole purpose is to actually create that replication fork itself. They're sort of like a pair of enzymatic scissors. So they're going to come in, and that enzyme is going to cut the hydrogen bonds that are holding these base pairs together. That's what these little lines are. Okay? And that's going to help to unzip that strand and open it up so that we can get access to the DNA itself, so that we can start to make copies. Once we separate those strands, these strands then can act as templates for us to copy the information. So if I know I have an A here, then I have to know that I have to bring in a new T to match it. Or a C here, then I have to bring a G in to match. And so they really serve as a strand, a template for us to make that new molecule, which is really kind of a clever way that DNA in and of itself is designed.
Alright, so here's the thing. Once that DNA helicase comes in and actually unzips the two strands, those parent strands, so these red strands here are the strands of that original molecule, before we can start building those daughter strands, or these blue ones, the enzyme that's going to ultimately be responsible for building those new strands is this enzyme called DNA polymerase. The problem is that there are some limitations to that DNA polymerase in the way that it can work. And one of those limitations is the fact that that DNA polymerase can't start adding DNA nucleotides to nothing. It needs something to bind to. It needs something to connect to. So the way that your cells have gotten around this problem is to actually build short strands of RNA nucleotides first instead, what we call these primers. And these primers may be 10, 20, 30, 40 RNA nucleotides long first, and then it can start building the DNA strand from there. So how does your cells do this? Well, they need yet another enzyme. And so that enzyme is called primase, and that's what you see right here. So that enzyme's job is to start adding RNA nucleotides together first. And the interesting thing is, for whatever reason, RNA nucleotides can start from nothing. They just need help from an enzyme to do it. So if I'm taking a look at building this daughter strand here, this blue one, that primase would have come in here at the end, used complementary base pairing to start building a short strand of RNA molecules. So they might be from here to here. Once we built that short primer, then that DNA polymerase can attach and start adding on from here. Those RNA primers provide us a starting point. Then the DNA polymerase is going to be able to come in and finish the job.
Okay, so helicase is the first one. That one comes in and zips open the strand so that we have a template to follow. The next enzyme that you need to know is DNA polymerase. And DNA polymerase's job is to actually bring in those new nucleotides. So if an A is here, DNA polymerase is bringing in the T to match it. Here's the problem, though, with DNA polymerase, is that your DNA molecules, those strands, run it in a very specific direction based on where the sugars and the phosphates show up in the backbone of that strand. Okay? And so when we talk about a 5' versus 3' end, the 5' end of that molecule means that that last nucleotide has its phosphate group unattached. 3' end means that its sugar has nothing attached to it. And if we look at this DNA molecule, these strands run what we call anti-parallel to each other. So one strand goes from 5', so a free phosphate, all the way down to a 3' with a free sugar. The opposite strand runs from 3', so the free sugar, all the way down to the 5' with the free phosphate. Okay? That's going to complicate this situation just a little, because your DNA polymerase can only add a new nucleotide to a free 5' end. Okay, meaning that that phosphate has to be available to bind the next sugar. So replication is going to happen on both of these strands at the same time, but the way that it happens is going to be a little different. Your lagging strand is going to be on this side, and that's going to be the one that runs from 3' all the way down to 5'. And that lagging strand I'm not going to be able to make continuously. My leading strand, however, runs from 5' to 3', and that one's running in the right direction. Okay, and so that one I can add new nucleotides one at a time all the way up the strand until I get to the other end. This strand, however, 5 to 3, I'm going to have to add in little chunks. All right, and so that's going to require a couple extra enzymes to help us here. We're going to have to use this molecule called primase, and primase's job is to add little snippets of RNA. Now I'm going to have primase happen once over here. Once that primase makes a single RNA primer, then I can make the DNA strand from there. I can use DNA polymerase all the way to the other end. Over here, though, I'm going to have to make little snippets of RNA and basically use DNA polymerase and work backwards. So I'm going to make little chunks at a time. Okay. Problem here is going to be that I'm going to end up with breaks. There's going to be pieces here that aren't going to be sealed together, the sugars to the phosphates in the backbone. And so I'm going to need to seal those together with another enzyme called DNA ligase. Okay, so let's recap for a second, make sure we're all on the same page. DNA helicase is one enzyme you have to know. Its job is to cut open the strands, cut the hydrogen bonds, so that we can have the strands open to use as a template. On my leading strand, so the one that goes from 3' to 5', that one's in the right direction for DNA polymerase. So I'm going to use primase to add a little RNA primer because DNA polymerase needs something to stick those new nucleotides to. And DNA polymerase, after that primer, is going to go ahead and synthesize that entire strand continuously. No problems. Your lagging strand, unfortunately, runs the wrong direction. It goes from 5' down here to 3' up here. So we're going to need to use primase to make little RNA primers, just like we did over here, and then DNA polymerase is going to work this direction to make a little strand called an Okazaki fragment. Polymerase comes in, makes another RNA primer, DNA polymerase then adds those bases again. Fortunately, there'll be a break here between the sugar and phosphate backbone, so DNA ligase comes in to seal that break. Okay, and those little chunks are going to keep happening until we get to the end of the lagging strand. So here's another way to look at it. Okay, here's my replication fork. Okay, so here's helicase coming in, breaking those hydrogen bonds. This side, running from 3 to 5, is my leading strand. We're going in the right direction. So primase comes in, it's going to add a little RNA primer, and then we're going to use DNA polymerase to keep adding new nucleotides until I get to the other end. Your lagging strand runs the wrong way. Okay, it's running from 5' to 3'. So we're going to need to use primase to make some primers, and then DNA polymerase works this direction to add new bases. Alright, so here's my primase, and then DNA polymerase works this way. In order to seal up these breaks, I'm going to use DNA ligase. Once that comes in, now I've attached a bond between the sugar and the phosphate from there. All of this ends up looking like what we call a semi-conservative process. What does that mean? Well, semi, kind of like half, and to conserve something is to keep it. So when I go through DNA replication, what I end up doing is taking that original strand, okay, and I use half of it, so let's use this dark blue one, that stays in that new molecule I create, and the orange one's the new one that I just made. In the other molecule that I create, here's the red piece, that's going to stay in that new molecule, and then this light blue one is the newly synthesized strand. So what I end up with is I take one molecule of DNA, I make two identical copies of it, but each of those new copies of DNA has half of its molecule from the old, all right, and half of the molecule is newly synthesized.
It's just beautiful, isn't it? It's mesmerizing. It's double helix exciting. You really can tell just by looking at it how sort of important and amazing it is. It's pretty much the most complicated molecule that exists, and potentially the most important one. It's so complex that we didn't even know for sure what it looked like until about 60 years ago. It's so multifariously awesome that if you took all of it from just one of our cells and untangled it, it would be taller than me. Now consider that there are probably 50 trillion cells in my body right now. Laid end to end, the DNA in those cells would stretch to the sun not once, but 600 times. Mind blown yet? Hey, you want to make one? Of course, you know, I'm talking about deoxyribonucleic acid, known to its friends as DNA. DNA is what stores our genetic instructions, the information that programs all of our cells' activities. It's a 6 billion letter code that provides the assembly instructions for everything that you are. And it does the same thing for pretty much every other living thing. I'm gonna go out on a limb here and assume that you are a human, in which case every body cell that you have, or somatic cell, in you has 46 chromosomes, each containing one big DNA molecule. These chromosomes are packed together tightly with proteins in the nucleus of the cell. DNA is nucleic acid, and so is its cousin, which we'll also be talking about, ribonucleic acid, or RNA. Now, if you can make your mind do this, remember all the way back to episode 3, where we talked about all of the important biological molecules, carbohydrates, lipids, and proteins. That ring a bell? Well, nucleic acids are the fourth major group of biological molecules, and for my money, they have the most complicated job of all. Structurally, they're polymers, which means that each one is made up of many small repeating molecular units. In DNA, these small units are called nucleotides. Link them together and you have yourself a polynucleotide. Now, before we actually put these tiny parts together to build a DNA molecule like some microscopic piece of IKEA furniture, let's first take a look at what makes up each nucleotide. We're going to need three things. 1, a 5-carbon sugar molecule, 2, a phosphate group, and 3, one of 4 nitrogen bases. DNA gets the first part of its name from our first ingredient, the sugar molecule, which is called deoxyribose. But all the really significant stuff, the genetic coding that makes you, is found among the 4 nitrogenous bases: adenine, thymine, cytosine, and guanine. It's important to note that in living organisms, DNA doesn't exist as a single polynucleotide molecule, but rather a pair of molecules that are held tightly together. They're like an intertwined microscopic double spiral staircase, basically just a ladder, but twisted, the famous double helix. And like any good structure, we have to have a main support. In DNA, the sugars and phosphates bond together to form twin backbones. These sugar-phosphate bonds run down each side of the helix, but chemically in opposite directions. In other words, if you look at each of the sugar-phosphate backbones, you'll see that one appears to be upside down in relation to the other. One strand begins at the top of the first phosphate connected to the sugar molecule's 5th carbon, and then ending where the next phosphate would go with a free end at the sugar's 3rd carbon. This creates a pattern called 5' and 3'. I've always thought that the deoxyribose with an arrow with the oxygen as a point. It always points from 3' to 5'. Now on the other strand, it's exactly the opposite. It begins up top with a free end at the sugar's 3rd carbon, and then the phosphates connect to the sugar's 5th carbons all the way down. And it ends at the bottom with the phosphate, and you've probably figured this out already, but this is called the 3' to 5' direction. Now it is time to make ourselves one of these famous double helices. These two long chains are linked together by the nitrogenous bases via relatively weak hydrogen bonds. But they can't be just any pair of nitrogenous bases. Thankfully, when it comes to figuring out what part goes where, all you have to do is remember that if one nucleotide has an adenine base, only thymine can be its counterpart. Likewise, guanine can only bond with cytosine. These bonded nitrogenous bases are called base pairs. The GC pairing has 3 hydrogen bonds, making it slightly stronger than the AT base pair, which only has 2. It's the order of these 4 nucleobases, or the base sequence, that allows your DNA to create you. So AGGTCCATG means something completely different as a base sequence than, say, TTCAGTCG. Human chromosome 1, the largest of all of our chromosomes, contains a single molecule of DNA with 247 million base pairs. If you printed all of the letters of chromosome 1 into a book, it would be about 200,000 pages long. And each of your somatic cells has 46 DNA molecules tightly packed into its nucleus. That's 1 for each of your chromosomes. Put all 46 molecules together and we're talking about roughly 6 billion base pairs in every cell. This is the longest book that I've ever read. It's about 1,000 pages long. If we were to fill it with our DNA sequence, we'd need about 10,000 of them to fit our entire genome. Pop quiz. Let's test your skills using a very short strand of DNA. I'll give you one base sequence, you give me the base sequence that appears on the other strand. Okay, here goes. So we got a 5' AGGTCCG to 3' and time's up. The answer is 3' TCCAGGC 5'. See how that works? It's not super complicated. Since each nitrogenous base only has one counterpart, you can use one base sequence to predict what its matching sequence is going to look like. So, could I make the same base sequence with a strand of that other nucleic acid, RNA? No, you could not. RNA is certainly similar to its cousin DNA. It has a sugar-phosphate backbone with nucleotide bases attached to it, but there are 3 major differences. 1, RNA is a single-stranded molecule, no double helix here. 2, the sugar in RNA is ribose, which has 1 more oxygen atom than deoxyribose, hence the whole starting with an R instead of a D thing. And finally, RNA does not contain thymine. Its 4th nucleotide is the base uracil, so it bonds with adenine instead. RNA is super important to the production of our proteins, and you'll see later that it has a crucial role in the replication of DNA. But first, biographies. Yes, plural this week, because when you start talking about something as multitudinously awesome and elegant as DNA, you have to wonder just who figured all this stuff out and how big was their brain? Well, unsurprisingly, it actually took a lot of different brains in a lot of different countries and nearly 100 years of thinking to do it. The names you usually hear when someone asks who discovered DNA are James Watson and Francis Crick, but that's bunk. They did not discover DNA, nor did they discover that DNA contained genetic information. DNA itself was discovered in 1869 by a Swiss biologist named Friedrich Miescher. His deal was studying white blood cells, and he got those white blood cells in the most horrible way you could possibly imagine, from collecting used bandages from a nearby hospital. God. For science, he did it. He bathed the cells in warm alcohol to remove the lipids, and then he set enzymes loose on them to digest the proteins. And what was left after all of that was this snotty gray stuff that he knew must be some new kind of biological substance. He called it nuclein, what was later to become known as nucleic acid. So Miescher didn't know what its role was or what it looked like. One of the scientists who helped figure that out was Rosalind Franklin, a young biophysicist in London nearly 100 years later. Using a technique called X-ray diffraction, Franklin may have been the first to confirm the helical structure of DNA. She also figured out that the sugar phosphate backbone existed on the outside of the structure. So why is Rosalind Franklin not exactly a household name? Well, two reasons. One, unlike Watson and Crick, Franklin was happy to share data with her rivals. It was Franklin who informed Watson and Crick that an earlier theory of a triple helix structure was not possible. And in doing so, she indicated that DNA may indeed be a double helix. Later, her images confirming the helical structure of DNA were shown to Watson without her knowledge. Her work was eventually published in Nature, but not until after two papers by Watson and Crick had already appeared, in which the duo only hinted at her contribution. Even worse than that, the Nobel Prize Committee couldn't even consider her for the prize that they awarded in 1962 because of how dead she was. The really tragic thing is that it's totally possible that her scientific work may have led to her early death of ovarian cancer at the age of 37. At the time, the X-ray diffraction technology that she was using to photograph DNA required dangerous amounts of radiation exposure, and Franklin rarely took precautions to protect herself. Nobel Prizes cannot be awarded posthumously. Many believe that she would have shared Watson and Crick's medal if she had been alive to receive it. Now that we know the basics of DNA structure, we need to understand how it copies itself, because cells are constantly dividing, and that requires a complete copy of all that DNA information. It turns out that our cells are extremely good at this. Our cells can create the equivalent of 10,000 copies of this book in just a few hours. That, my friends, is called replication. Every cell in your body has a copy of the same DNA. It started from an original copy and will copy itself trillions of times over the course of a lifetime, each time using half of the original DNA strand as a template to build a new molecule. So, how is a teenage boy like the enzyme helicase? They both want to unzip your genes. Helicase is marvelous, unwinding the double helix at breakneck speed, slicing open those loose hydrogen bonds between the base pairs. The point where the splitting starts is known as the replication fork. It has a top strand called the leading strand, or the good guy strand as I call it, another bottom strand called the lagging strand, which I like to call the scumbag strand, because it is a pain in the butt to deal with. These unwound sections can now be used as templates to create two complementary DNA strands, but remember, the two strands go in opposite directions in terms of their chemical structure, which means that making a new DNA strand for the leading strand is going to be much, much easier than for the lagging strand. For the leading, good guy strand, an enzyme called DNA polymerase just adds matching nucleotides onto the main stem all the way down the molecule. But before it could do that, it needs a selection of nucleotides that fill in the section that's just been unzipped. To get started at the very beginning of the DNA molecule, DNA polymerase needs a bit of a primer, just a little thing for it to hook onto so that it can start building the new DNA chain. And for that little primer, we can thank the enzyme RNA primase. The leading strand only needs this RNA primer once at the very beginning, then DNA polymerase is all, I got this, and it just follows the unzipping, adding new nucleotides to the chain continuously all the way down the molecule. Copying the lagging, or scumbag strand is, well, he's a freaking scumbag. This is because DNA polymerase can only copy strands in the 5' 3' direction, and the lagging strand is 3' 5'. So DNA polymerase can only add new nucleotides to the free 3' end of a primer, so maybe the real scumbag here is the DNA polymerase. Since the lagging strand runs in the opposite direction, it has to be copied in a series of segments. And here, that awesome little enzyme, RNA primase, does its thing again, laying down an occasional short little RNA primer that gives the DNA polymerase a starting point to then work backwards along the strand. This is done in a ton of individual segments, each 1,000 to 2,000 base pairs long, each starting with an RNA primer. These are called Okazaki fragments, after the couple of married scientists who discovered this step in the process in the 1960s. And thank goodness they were married so that we could just call them Okazaki fragments instead of Okazaki-someone's-someone fragments. These allow the strands to be synthesized in short bursts, and then another kind of DNA polymerase has to go back over and replace all of those RNA primers, and then the little fragments gets joined up by a final enzyme called DNA ligase. And that is why I say that the lagging strand is such a scumbag. DNA replication gets it wrong about 1 in every 10 billion nucleotides. But don't think your body doesn't have an app for that. It turns out that DNA polymerases can also proofread, in a sense removing nucleotides from the end of a strand whenever they discover a mismatched base. Because the last thing we want is an A when it would have been a G. Considering how tightly packed DNA is into each one of our cells, it's honestly amazing that more mistakes don't happen. Remember, we're talking about millions of miles worth of this stuff inside us, and this, my friends, is why scientists are not exaggerating when they call DNA the most celebrated molecule of all time. So you might as well look this episode over a couple of times and appreciate it for yourself. And in the meantime, gear up for next week when we're going to talk about how those 6 feet of kick-ass actually make you you. Thank you to all the people here at Crash Course who helped make this episode awesome. You can click on any of these things to go back to that section of the video if you have any questions. Please, of course, ask them in the comments or on Facebook or Twitter.